Good afternoon, everyone. My name is David Regan. Thanks for waiting for a minute or two, just sorting out a number of things. Very excited to have you with us today for this webinar for investors. Excited about some of the things we have to talk about today. What we're going to tell you about today is the culmination of a great deal of work that builds on both our past preclinical and clinical studies that launches us into the future. What we're sharing today is our forward clinical strategy. And what you're going to hear about is a combination approach that really plays to Sona's strengths. And we're going to share with you the thinking, the logic, and the rationale that goes into why these particular studies were chosen and why we're so excited about where we think they can take us. As many of you know, I've got a bad echo, I'm being told here too. So I don't know if that's on a single line or on all lines. We didn't have that in the test room, so I'll continue. The immunotherapy industry, and what we're going to be describing today, is a combination therapy of our targeted hyperthermia therapy with immunotherapy. Immunotherapy, of course, marked a major advancement in cancer care 15 years ago, and it is now a massive industry with a massive problem facing about $300 billion in revenue that have patent cliffs facing them in the coming 10 years. That said, that also can present opportunities for some. So today we're going to share with you our plan to start transforming the oncology industry. But first, of course, we point to our forward-looking statements and refer you to our disclosure documents because we'll be making a number of different comments today that relate to forward-looking statements. And so we add those cautions as always. Now, today we'll have a brief background for those who aren't familiar with things. Carman will then share our insights to date, what we have learned effectively, and then we'll launch into the new studies and finally describe where this is all going. However, first, I would like to welcome our guest panelist, who we are truly honored to have with us. And his timing couldn't be better because I think he's just chiming in at this very moment from overseas. So Dr. Michael Smylie is a true luminary in the oncology world, and particularly in melanoma. So we're very fortunate to have him and look forward to him chiming in as we go through the presentation, and we'll help with some of the Q&A. On the point of the Q&A, of course, there is a chat box in your Zoom that you can feel free to click on and add questions in, and we'll get to them as is appropriate through the course of the webinar and after. So, as we were saying, immunotherapy was a major breakthrough in cancer care, and it has done an enormous amount of good, but it has, in the case of melanoma, typically helping less than 40% of the patients who are administered those drugs. And across different cancer types, in one particular type of immunotherapy, a study commissioned by the manufacturer, showing an average of 21%. What this means is there's just a huge opportunity for improvement here that we need to. This is an industry and a drug that has been nearly fully played out now. Lots of commentary on that, talking about how as they've reached this plateau, new combinations of the existing immunotherapies have been tried to great expense and to generally little effect. So it's our goal to break through beyond that plateau. Plus, of course, immunotherapy has considerable toxicity associated with it, in some cases 15 to 20% grade 3 or higher. It's very unusual for a patient to make it all the way through without some significant side effects from those immunotherapy drugs for all the good that they do. So the central question we're here to address is, can we improve on this at the same time as reducing the toxicity? This is the problem in the world that we've been set up to answer, and we're going to take you through why we think we can do it. So Sona Nanotech at its core makes gold nanorods. We are the only ones who make biocompatible gold nanorods that are toxin-free in their manufacture. Gold nanorods are important because they're highly functional. They have a length and a width, so you can tune them to wavelengths makes them functional for in vivo applications. They're, as I say, uniquely biocompatible, and they've been validated by the Nanotechnology Characterization Lab in the US. Most important for our purposes, beyond the biocompatibility, is the fact that they are the most efficient nanoparticle at converting non-thermal light energy into heat. And that is exactly what we do with our targeted hyperthermia therapy. We take, as you can see on the left, immune cold tumors, ones that are not responding to immunotherapy, and we effectively prime them, which engages the immune system. We prime them by injecting gold nanorods directly into tumors and then applying our light, our near-infrared laser light on them, bringing the temperature up to about 45 degrees, which is hyperthermia, that is subablation. That's an important temperature because at that stage, we are killing cancer cells, but not harming healthy cells. And of course, all of that generates a tremendous amount of immune activity. And what we have seen is that immune activity turns those previously immunogenetically cold tumors hot, pardon me. And this is what we're doing to it. It's great effect from our first in human study. The photo on the left before we started treating major tumors, many minor tumors. This is a late stage melanoma case, but after only 2 weeks, on the right, you can see that the treated tumors are healing. And perhaps more importantly, the secondary untreated tumors are diminishing. And that is what we call is called the abscopal effect. And that is something that we're going to be pursuing in these studies as well. So in our first in human study, we had out of 10 biopsied tumors across 10 patients, 8 that showed that they had in fact engaged the immune system, that there was an immune response that wasn't there previously. We did that with biopsies. And in 6 of those cases, after 2 weeks, there was no evidence of cancer in the tumors, and that's significant. Finally, the third item on the bottom right here is our histology study. After the fact, some of the follow-up data that was done demonstrated and proved the extent of immune engagement that was significant. And with that, I'm going to turn it over to my colleague, our Chief Medical Officer, Dr. Carman Giacomantonio.
Thanks. So what we learned from the clinical trial and from all the preclinical work has really paved the way to what we're about to embark on. So the next slide, please. So the first clinical study gave us the first bullet. So T primes tumors. David just mentioned that. So, and that's what it does. So it's priming tumors. What does prime mean? Prime means the cold tumor, the tumor that's invisible, now becomes visible, A, because it's giving off signals that it's there that it didn't do before to the immune system, and B, fever heat. That's essentially what this is a high fever. Fever heat focused into a tumor draws in immune cells to the area of concern. The second thing that we demonstrated was that 44 degrees is correct. So when we started, we didn't, we wanted to be subablative. This is subablative, meaning that when we treat the tumor, we're activating cellular death rather than killing tumors through heat. It's a very different process. 5 minutes is enough. That's very important. When it comes to an application of a treatment that you're going to take into the clinic, can you do it quickly? Because that's how efficiently and quickly can you get a patient in and out and thereby minimize the impact on them. The next, so two treatments with THT. This wasn't intuitive. This is what our pre-treatment, our preclinical work showed us. When we did 2 treatments, we got a very long window of immunity activated in those tumors. And so to our surprise, and the next bullet, what we showed was that while our preclinical models, we showed us that after a few weeks, we started to have regrowth. Several of the patients we treated in that clinical trial didn't experience regrowth, even at 6 months when we just reviewed their data and are now preparing the manuscript to submit for publication. The other thing that was really important was as I indicated above, that 5-minute issue, staff can learn this. So this can be done in a clinical setting, doesn't require a hospital, doesn't require radiation bunkers and gantries and things. It requires very minimal clinical support with skilled staff, of course, to treat the patients. But the requirement for the supportive equipment is so minimal that it makes it it opens this up to so many patients that otherwise wouldn't have access. So our preclinical work carried on, of course, while we were doing our first clinical trial. We showed that THT, so the target hyperthermia, through priming the tumor, actually improves the performance of immunotherapies. And this is a study we just published in April. And we took this very cold colorectal cancer tumor model, and we treated them, as you can see there, with no treatment, meaning that we gave laser, but no other treatment. When we added the immune therapy alone, we didn't see any tumor response. And then when we did targeted hyperthermic therapy and added the immune therapy, we got a complete response. And so this model, and I can, for some of you that are involved in science, will agree that it is rare to see such a flattening of tumor in these preclinical models. So with that insight, with that background, with our experience both in the preclinical model, showing that we can not only activate, but make the immune therapies more effective, and our clinical study showing us that we can do this safely in patients, it's time to combine the two. So onward we go, combining THT with immunotherapy. So what's the driver? We know it primes the tumors. We've shown it in the preclinical model. And I will add, we've shown it in the clinical experience. The multiplex staining that we did showed, and you will see as the publication comes out, that the immune cells activated through targeted hyperthermia therapy were just what the tumor needs, and in fact, what an immune therapy needs to be effective in the tumor microenvironment. The second thing we showed was that the preclinical model showed that we make immunotherapy response rates exceedingly high. And so this is optimal. We believe this is as optimal as it can possibly get. And then the third thing is we showed that it's well tolerated. And that's critical for this. It's critical that we can do this 2 short therapies, 5 minutes of treatment itself, 15 minutes per lesion is what we can show you, and we'll show you in the paper. This is extremely well tolerated and easily delivered, relatively speaking, therapy. So this is compelling background. We feel comfortable that we've de-risked the situation as best we can, and we're optimistic that we will see excellent accrual and good responses. So what's the current state of the art? So current standard of care for immunotherapy is stage-based. What I mean is this: in stage 1 and 2 cancer, so cancer, melanoma that's localized to the skin, hasn't spread to the nodes or beyond. Stage 3 and 4 is cancer that's spread to the nodes and beyond. For the most part, the standard of care is if you have a stage 1 or 2 cancer, you'll go see a surgeon, a plastic surgeon, a dermatologist. They will surgically remove the cancer, and that's the end of it. For the resectable, meaning that it's surgically clearable, stage 3, with newer trial results and evidence, we will give immunotherapy with the surgery either before or after, and we can talk about that. And then for the unresectable, we give immunotherapy in the hopes that the tumors will respond, and we can either clear the disease or change them to something resectable that surgery can help with. In the past couple of years, there's been an evolution of the treatment. So now, the next slide will show we're moving the immune therapy into the stage 2 area. Now, as I said, stage 2 is a lower risk, but there are certain high risk features of stage 2 that show us that they have even maybe more risk of spreading to the body beyond the lymph nodes than some of the stage 3s. And so because of that, trials have been done and completed to show that if we give the high risk stage 2 patients immunotherapy with their surgery or after their surgery, that we can improve their outcomes.
Can I ask a question, Carman?
Sorry?
May I ask a question?
Sure, please.
Why isn't immunotherapy used for resection in 2A for stage 2 cancer?
Well, that's a good question. And so, the biggest reason is because of the slides you showed before about the toxicity. So an immune therapy might have efficacy in a low risk stage 2, but it is low risk, meaning that the surgical intervention itself is generally sufficient to cure the disease, and so the toxicity is limited to the surgical treatment. If we add an immune therapy that has a 15% risk of severe stage 3 or grade 3 toxicity, then the toxicity doesn't justify the benefit when surgery was probably adequate. So we really, the more we de-risk anything we do in addition or instead of surgery, the more likely it will be added to or replace surgery.
Great.
So now we're moving. So this is the stage we're at now. So we have shown that we can ignite, we can activate immunity in tumors that were previously cold, that previously didn't respond to immunotherapies. So the target population, going back to our first clinical trial, which were people who were unresponsive to immunotherapy, we showed that we can take the most resistant diseases and activate immunity. Now what we're going to do is go into the population with people with the similar stage of disease, i.e. unresectable or not resectable with significant trauma or surgical concern, and we're going to treat these patients with our targeted hyperthermic therapy in combination with an immunotherapy. The premise being that we have activated immunity through the THT that would not be accessible with standard immune protocols. So this is kind of a summary of the IGNITE. The IGNITE is the acronym. This is intratumoral immunotherapy plus targeted hyperthermic therapy to generate novel immune tumor eradication, i.e. IGNITE. This is an early feasibility study similar to the last one. Now that we're combining an immunotherapy with the targeted hyperthermic therapy, we have to be cautious to make sure that the combination of the two in the human experience will be tolerable and acceptable. So we're targeting, again, those stage 3, the advanced stage 3 and stage 4 melanoma patients. In this case, these patients will not have seen immunotherapy. They will be entering into the system. So, of course, they will have to receive a standard of care. So they'll be screened. They'll have to be healthy enough to undergo treatment that's potentially curative. And then they'll be entered into a standard immunotherapy. Standard immunotherapy in many centers in many countries is just PD-1 inhibition. Remember, PD-1 stands for programmed death receptor. It's a target. It's the commonest target of immunotherapies in today's world. But concurrently in this group, we will treat them with one of 3 arms. So arm 1 would be intralesional IL-2, so directly into the tumor, interleukin-2. It's a standard of care in Canada, so we would be held to that standard. Patients would have to see if that makes the difference. The second arm would have interleukin-2, our standard, plus our targeted hyperthermic therapy. Our preclinical data tells us that the interleukin-2 will have far better effect in the tumor, the immunotherapy, once we've treated it with THT. The third arm is a very exciting arm in that we would treat it with our targeted hyperthermic therapy, and we would add 2 immune agents. The interleukin-2, which is, again, the standard, and this other one, CTLA-4, it's a targeted checkpoint that attacks regulatory cells, so it turns off the regulators. So
another immunotherapy.
Another immunotherapy.
So 3 immunotherapies at play here in combination with THT.
That's right. So this, this kind of therapy, so what's expected is if you combine these things, it's based on everything we've seen today, we should see a far better response. We should see increased local control. We saw that with our just targeted hyperthermic therapy alone, far better local control, and we should see decreased treatment-related toxicity, which is, of course, the goal. If we're, as David mentioned earlier, if we're combining immunotherapies, what we've shown, and when we do it systemically, we give them both in the veins, that the toxicity goes way up, and the benefit, there's a plateau. The benefits don't justify the toxicity. If we are separating them and focusing some of that treatment into the tumor as we do our targeted hyperthermic therapy, the sound rational hypothesis is that we have minimized the toxicity or the exposure to the person and maximized the exposure in the tumor where the immunity is needed. So what will IGNITE treat? IGNITE is going to address the worst of the worst, the highest of the risk. And so this is laudable. That's exactly what we want. The stage 4 patients, the stage 3 patients, they're going to be on our target. But of course, that's the tip of the iceberg in melanoma. There's so many more patients that we believe we can, just as immunotherapy moved into the stage 2 patients with the highest risk, we believe we can improve those results and reduce the amount of toxicity associated with the improvement. So this is really the target. So to this point, to prove that we can make bigger impact in that earlier stage group, our other trial will be the PRIME trial. So the PRIME trial, essentially, this is upfront, in the jargon, neoadjuvant, but upfront combination therapy with immunotherapy and targeted hyperthermia therapy given. And the idea is this. I'm going to just digress for a second. When we started treating stage 2 patients without nodes with immunotherapy and surgery, we gave the immunotherapy because even though we knew the surgery would cut away the disease, there was a significant risk that those patients would recur where they started or the disease would have spread. So what we're trying to do is improve those issues. Can we reduce the local recurrence rate and prevent the spread of that disease beyond in combination with surgery? So we are looking at the stage 2-3 group, the same kind of group that we looked at with where the immune therapy has shifted before, where we believe we can show that we can improve the local response rate and reduce the distant disease rate and reduce the toxicity of the immunotherapy by treating part of the immunotherapy in the tumor or delivering it into the tumor. So the next slide explains it a little bit more and just elaborates on it. So again, PRIME. So yeah, so exactly. So the PRIME. So PRIME is precision regional immunotherapy for melanoma enhanced by targeted hyperthermic therapy. So PRIME-THT. So again, this is an early feasibility study. We have to be cognizant of the fact that we haven't done this combination, and so we want to prove that we're safe to doing that. We're going to start with 10 patients. These are stage 2, stage 3 patients, so surgically resectable but have high risk. They will be started on the current standard of care in a upfront treatment, which would be an immunotherapy systemically, and then these patients will then receive, at the same time as that's initiating the systemic therapy, intratumoral targeted hyperthermic therapy plus our immune combination. We're giving this triplet therapy because we believe that this is the most effective first line in treatment, and giving the other 2 immune agents in the tumor will minimize the toxicity that could be seen if we were to give those both systemically. There is a trial, I can tell you, that's been done with that CTLA4, one of the immune therapies, and the PD-1 inhibitor, and it's had a very good response rate in these patients, up to 60%. It's had some significant toxicity, and it takes a long time. We believe we can improve that response rate by another 20 to 30% and reduce the toxicity by taking away one of the agents. After the 3 treatment, sorry.
And yet, I was going to say, what if the study only produces the same efficacy as that combination immunotherapy, but without the toxicity? Is that not superior?
Well, so that's a good question, and I'm going to ask Michael to comment on that in a second. But we call that, and these studies are done as non-inferiority, meaning if it's at least as good as the standard of care with less toxicity, it's more desirable. And so what we would be looking for is at least as good a response as the current standard. But if the patients have fewer side effects, that would be something that would be very appealing to both treaters and patients alike.
And payers.
So, and then based on the other trial that was done, the Pareto trial, this at 42 days, which is a standard of care, by the time you get diagnosed from a dermatologist, you get sent to me or a plastic surgeon, and we do the surgery. This is the natural window. If we add pre-treatment immunotherapy, which is the standard in North America, that's given for a couple of treatments, and then there's a little wait period before you go. So this is well within an accepted standard of time from the diagnosis to surgery. And then this trial is designed as an upfront treatment. So these patients are going to go on to standard of care, which is to excise, resect, remove surgically their primary tumor and any lymph nodes if they were involved, and we would know that before we started. And then that will give us immediate information. Did we effectively treat the primary tumor where the disease started? Did we effectively have an impact on any disease that was in the nodes? And based on those results, the PI of the study will determine whether or not we carry on with further standard of care treatment, which would be more intravenous systemic immunotherapy, depending on disease residual. Now, Dr. Smylie is a medical expert and is actually one of the founders of this kind of research, and so he would be best to comment on why we would or wouldn't do that, but we can get to that. So what we expect, though, with this is that based on everything, the last clinical study, our preclinical model, we should get far higher local response rates than what we're seeing with standard care now. The local treatment, of course, is important, and that's what this is, because we're going to treat it surgically anyway. So I already said we're going to clear it one way or the other, but it's the effect in the lymph nodes and beyond that will tell us really how effective this upfront therapy was.
And Carmen, there's a question here saying, doesn't preventing cancer spread take years to assess? But you're saying there are near-term metrics as well.
Yeah, that's 100%. And so that's a very good question. That's important. So in order that it takes time. So, but there are correlates of or predictors of outcomes. And so the higher the upfront, the higher the response in the tumor we treated and in the lymph nodes, predictably, the fewer recurrences in those nodes and beyond we'll see. Now time will have to prove that out. So that's a fair question, and so we believe this study. But if we see the equivalent effect to the neoadjuvant, the upfront treatments that we're seeing now with less toxicity, we're at least on safe ground to say that this is as good or even a better alternative for patients.
So, melanoma is sadly a growing market, both in terms of numbers and the value of what's being spent on therapeutics. That's projected by Grandview Research to grow to $10 billion a year within the next few years on melanoma. It's a significant market that encompasses all therapies that are being applied today. We think this is an important place for us in that we can bring enormous value to patients, to the people funding the medical system, and to the potential partners who have big vested interests in seeing benefits accrue to these patients and are active in those markets currently. So a little word now then on these proof of concept studies, we call them that. It's an early feasibility study, but it's going to provide the evidence for these two very, very novel therapies relatively quickly and cost effectively. And that's important from an investment perspective because it provides a lot of torque on this stock. So walking through the clinical strategy we've just defined, there are effectively three studies we are pursuing: the IGNITE-THT study, the PRIME-THT study, which is early stage melanoma, and finally the larger scale clinical trial in Canada that we continue to work on, which will be the culmination of this. These are interesting studies, and I'll let perhaps Dr. Smylie comment on some of that. Success metrics here, and there's some questions on this too, but success metrics are, of course, safety and tolerability. Yes, we've already done one first-in-human study, but that was THT alone, really to show that that is safe. Now, by bringing in what is effectively a new therapy, we've shown one part is safe. We have to show the second part is safe too. The phasing of approvals is always stage gated by a small number of patients and then growing as safety is shown. As I think many know, that the biocompatibility of our therapy lends itself, and as Carmen highlighted, we showed excellent safety and tolerability in our study. These are proven and approved immunotherapies. So we have every confidence that we're going to hit safety and tolerability in the same fashion. And so the next question is going to be tumor response. And for IGNITE being for late stage melanoma, it's local, regional, and distant, that abscopal effect that we're looking for, that we saw evidence of when we treated patients alone with THT. But when we bring in the immunotherapy to sustain the effect of THT, we're really excited to see how much further we can show that. And of course, the durability, which is a time factor. And we'll talk a little bit about the timing of this. But the first indication is, did we prime the tumor? And then it's a question of, did the tumor fade away, and did it stay gone? And that, of course, is an ongoing assessment. Some of the most compelling information from our last study came with the 6-month follow-up. In the case of the PRIME-THT study, which is early stage, it's going to be the same safety and tolerability, the durability and tumor response, both local and regional. And Carman, do you want to speak to that in terms of the why those tumor responses are different, the metrics?
The local and regional, the local response is when we're doing anything with a intratumoral therapy. The local response has always multiple factors that it could influence. That. So the volume of the drug that's concentrated in the tumor, the amount of heat we deliver. There's a whole range of factors. So a good local response is very encouraging. The local response that we're really interested in is how robust is that immune activation? What do the cells look like that are going in? The regional response is equally important. And so with the PRIME and the IGNITE, where we treat one tumor and we see nearby or regional tumors in the lymph nodes or beyond the range of where the immunity that we initiated by treating the tumor would be expected to be, that's an indication of a systemic response. That means the immune cells the immunity is actually started in the tumor, but actually activated in the lymph nodes around the tumor. And that immunity has to circle back through the body's circulatory system to get to where the tumor is. And in doing so, has the opportunity to encounter other tumor-like cells that bear those signals that we're looking for, the cancer messages, the cancer antigens. And so so that's local, that's regional, and that's distant, because there might be a tumor in a lung or in a bone that looks the same. And because the blood distributes throughout the entire body, those immune cells will be in that blood that's being distributed and has an opportunity to see that tumor in a way it didn't see it before. All of that leads to this. So somebody asked a very good question. It takes a long time, but it doesn't take a long time when there's disease present. So if there's disease in a node, the response is quite quick. What we don't know is if there was disease somewhere else, not in another node or outside in the body, that we couldn't see or measure. Now we only have time to tell us, because it's beyond the sensitivity of any tests we have right now. In our clinical trials, we're going to do fancy blood tests to see if there's evidence of circulating tumors and things as well. And while they're very reassuring, nothing is perfect yet.
So that's almost more difficult to do, doing it early stage, because there wouldn't be necessarily evidence of any spread yet.
No. But again, as I had indicated, and I'd ask Michael to comment on this, there are correlates. And in the medicine, all throughout medicine, we look for correlates. Response in one area or a type of response can be often an indicator of what is going on beyond, even if you can't see it or appreciate it at this point. I wonder, Michael, are you able to and thank you for being here today. Michael's in Germany, so he's really going out of his way to help us today.
No, that's fine. Can you hear me okay?
Yes, thank you.
Perfect. Okay. So do you want me can I make a comment just about a couple things that came up? So
please.
Somebody just asked what CTLA-4 was. And basically what I always tell my patients, that there's a priming phase and effector phase, and CTLA-4 works at the level of the lymph node where the cells are presented with the cancer cells. And then once they recognize it as foreign, they start to produce other T cells that will travel out and attack the tumor. And you have to have brakes on the immune system, because you never want too strong an immune response. So as soon as the immune system is turned on, it starts to turn off. And one of the first brakes that is expressed on the immune cell is CTLA-4. So by blocking that, we can get a more sustained proliferation of T cells that will move out and go to the tumor. More importantly, more clones, so different types of T cells that will recognize different parts of the cancer cell. The PD-1 inhibitors are basically work at the level of the tumor. So they're like the offensive lineman on a football team. That it's a localized break, and cancer cells can manipulate that break on the. So as soon as the T cell is turned on, it starts to express PD-1, and T cells can or cancer cells can mimic another immune cell and activate the PD-1 on immune cell and function to turn off the immune cells. But so by blocking that PD-1, we can reactivate the immune cells, and the immune cells hopefully will then turn around and start attacking the cancer again. The second question that came up is how long do you have to wait to see the long-term results about and whether cancer is going to recur? Now, we had these discussions very early on when we started using immunotherapy too. And the difference with immunotherapy versus chemotherapy is that when you give chemotherapy, you get a sort of exponential cell kill, but you never get to zero, and the curves never really flatten out. But with immunotherapy, you see the curves go down, and in about 3 years, they plateau, so they go flat. And that's very important because when the curve flattens out like that, that tells us that percent of patients are getting long-term survival. With ipilimumab, it was about 20% long-term survival. With PD-1, it was about 33. And then when we combine the two, that at 5 years, over half the patients are still alive. And this is a cancer where just 5 years earlier, the median life expectancy for patients was 6.2 months. Now it's 6 years. So we've made tremendous advances, but saying that, still half the patients ultimately die of this disease, so there still is an unmet need. The third question that came up is just why would we use, when we get such good results with local therapy, why would we use something different? And as Carman said, that the immunotherapy carries with it substantial toxicity when you use ipi and nivolumab, a PD-1 together, that about 60% of patients will get grade 3 to grade 4 side effects, which are serious to life-threatening. And so it takes a team of experts to really manage those side effects. And some of those side effects, such as the endocrine ones, so type 1 diabetes, which can occur, pituitary failure, so you can lose your cortisol production. They're life-changing side effects, and those ones are irreversible. So I think there's even a push now for some people to stop funding these immune checkpoints in stage 2 disease, because they say that, as Carmen said, that the earlier the stage of disease, the lower the risk of recurrence. And then they're questioning, well, do you really want to give people a toxic immunotherapy that might give them long-lasting side effects if they're not going to relapse at all? So I think these localized treatments that they've been talking about are very important to try to get, decrease the systemic toxicity and get good local control over their disease.
Yeah, that's excellent. Thank you, Dr. Smylie. That's really helpful. And there's a question here actually asking what role Dr. Smylie might play in IGNITE and PRIME in these studies. Carman, do you want to speak to that?
Yeah, so, and that's an excellent question. So Dr. Smylie's already been involved, and so his experience in developing these therapies back with the big pharma years ago, he has a great sense both of what toxicity and what efficacy or what effect these things can have. And so he's been instrumental already in advising us around some of the protocols. So for instance, patients in our studies will receive a systemic immune therapy in conjunction with our intratumoral therapy. And the reason is there's a standard of care that we have to uphold. And what the immune therapy that we're offers a shorter course, which translates often to lesser experience with toxicity, a shorter exposure to the patient. So the other role is like all of us at Sona, Dr. Smylie is come on as a scientific advisor. All of us, we design the studies, and Dr. Smylie is, again, a medical expert, and there's a big, heavy medical arm to this. And so his sage advice is not only welcome, it's necessary. But once the study design is finalized and approved by the ethics boards, then it goes on to the principal investigator, and we are actually at arm's length beyond that. We are not allowed to interfere, to redirect, to adjust. In a feasibility study, we are given opportunity from time to time, particularly if there are things that are not feasible, for us to pause the study and adjust and reintroduce. But that's the extent of it. We are not allowed to influence the selection, the treatment. So the treatment algorithms are predetermined, and then the study is run according to those algorithms. And any deviation from a treatment algorithm is a big deal and requires a committee review. And so Dr. Smylie is instrumental in assuring that the likelihood of that happening in one of our studies is low.
That's it. That's it. So the next thing we'd like to talk about here is then on the timing of that. And of course, there are two factors on timing. The number one for any clinical study or trial, it's the enrollment period is the driver of the length of time. How quickly can you attract, secure the patients who clear the eligibility criteria and are willing to participate to meet the numbers that we need. Now, in the case of IGNITE, it's a larger study, in this case, with 30 patients across the 3 arms, 10 in each of the arms. In the case of PRIME, it's 10 patients. So PRIME, we expect to be a faster enrollment as a result of that. And in fact, what you'll see on enrollment being the first factor, sorry. Here we go. There we go. The enrollment period. Well, there's the enrollment period, and then there's the early readouts period. So enrollment for IGNITE may take up to 12 months, given the numbers. It's for this reason that we're going back to Chile, where that has a very high incidence, much higher than Canada, incidence of melanoma to be able to accrue as quickly as possible. We expect to have early readouts much more quickly than that. In the case of the PRIME study, it's a faster enrollment period. It's largely the same patient pool, one early stage, one late stage. As we saw with some of the graphics earlier, the early stage has a much bigger population, about 10-to-1 ratio. So that could be, in fact, quite quick. And then early responses are going to be inside these periods as well. In the case of the Canadian larger scale study, both of these studies will inform that study. Now, that's the study that we are engaged with Health Canada on right now. They've shared questions with us. We are addressing those questions. There are no questions that they've provided that we don't believe we can address, but we also can't speak to timing in terms of, we can't speak for them. That's not appropriate. That's in their court. We continue to press as quickly as possible in that front, but we see that, as we've indicated, as starting up. We're gauging now for January with that, and the benefit of that, of course, is that we'll have early results from both of these trials to, number one, inform that trial and the probability of its success, as well as raise the necessary funds for it. Because by going to Chile, we can again do this highly efficiently, working with the same clinic, same principal investigator, who is eager for us to continue for multiple reasons in Chile, and we'll be able to do it providing a great deal of torque when we can provide results by this fall in such a quick window, taking us from today, where we have had 10 patients on a monotherapy with THT, to this fall with the true combination therapy that we think can be so powerful, both in raising the efficacy of immunotherapy and reducing its toxicity and showing real durability to it. Now, the durability aspect of it is going to take some time, where we want to measure that out over a number of years, but we'll be able to assess significant responses immediately. And as far as, we get questions also on funding it. Obviously, we're an R&D company. We're public, and we tap the markets occasionally when needed. And that said, we have, numerous things we're working on right now, and we're going to do what is in the very best interest of the company. And we're very confident that we've got some really exciting studies that are highly investable right now that we're sharing with you today, that will have results before the end of the year in terms of early readouts. So we're excited and think that's got a strong future in front of it. Now, where else is this going? So this clinical strategy is creating value. It's creating scientific validation for us across a number of very, very novel approaches in terms of the early stage approach of that could possibly even take immunotherapy to where it hasn't typically been done early stage. It's a risk-balanced expansion. Look, when we saw how impressive our first study's results were, there were some who came to us and said, look, you got to go after pancreatic cancer. Well, that is a Hail Mary that this company's not willing to take at this stage. We will love to get there, and there are many cancers many solid cancers we think we can address. But we know that this has worked in melanoma to date, so we want to build out in a fashion that has minimum risk and maximum upside, which is what we see by continuing to go deep into melanoma and show the true power of immunotherapy in sustaining what we start with our THT. Also, partnership appeal. Partners want to see not that you've done a little bit on this cancer and a little bit in that cancer. They want to see depth. And that's exactly what we get with these two studies coming out. Regulatory momentum is important, and that's why the Health Canada discussions continue, and we push on there. And we may be surprised and have something earlier, but we've learned to be cautious in that regard. And regardless, there are multiple near-term valuation catalysts that we'll walk through. In fact, in the next slide, perhaps. So the path forward specifically takes us to the next thing is that Carman's manuscript from the Chilean first-in-human study is being submitted for review scientifically, and then he'll be sharing that at the American Society of Surgical Cancer Oncologists. That's it, ASCO, the largest gathering of oncologists in the world. Which is heavily favored by industry, and will be very excited to both publish those results and share them with that oncology world. These two combination therapies, one for late-stage melanoma, show the durability, and one for early stage that will show our ability or not to prevent the spread. That can have some really profound impacts, both in terms of the early readouts and the later readouts. And of course, the clinical trial in Canada that we continue to move towards. Strategic partnering, lots of questions around that. Every partner wants to see human data, and then they want to see more human data. And I promise you that we are sharing that, and getting it published is job number one. That data effectively doesn't exist in the industry until it has been peer-reviewed and published in a reputable scientific journal. So that is job number one that leads towards the partnering conversations. And finally, the research and IP development continues in the background. We continue to build out our moat here, and we feel like we are on the leading edge of this technology and this movement, and it's important not to stand still, but to move forward. And so there is work going on in the background. Carman talked about how our preclinical work informs and facilitates our clinical work. So for future indications of cancer that we go into, we'll continue doing that work, and we'll continue filing patents when and wherever we can. That is what we have today, but I want to see if there are any more questions here. We're asked, when is the ASCO conference? ASCO conference, Carman, you're presenting what, June 2nd?
Yeah, actually, end of last May 30th, I think. Yes, it's that weekend.
Okay. End of the month. And a question in terms of, are we confident we're staying ahead of the competition? We are right now. We've got, you can view the competition quite broadly in terms of immunotherapy. And the goal of these two studies is to show how much better we are than standalone immunotherapies. And we have an incredibly high degree of conviction because of what you've seen and because of what we've done. Every study we've done has produced tremendous results and strong science, and we have every confidence that will be the case in this case, and that helps us against that competition. Now, there are hundreds of developing concepts to address cancer, and we believe we're towards the front of that, and we need to stay at the front of it, so we need to keep working and keep building out the science, showing the amount of data, building the data, and having more human results. I want to turn it over to see, yes, if there are any other questions, because I want to certainly thank Dr. Michael Smylie, whose comments are very helpful. But any other points you wanted to make, Carman?
Yeah, so no, I think the points are made. They're very good questions, and a lot of the questions are unanswerable. Do we have interest from industry and partners? I can tell you, we were just at the AACR, the American Association of Cancer Research. There's lots of interest from industry types. People are, as David said, and I'm going to ask Michael to comment on this too. We have reached a plateau in immune therapy, and so where we have to be innovative to raise the response rates above those current rates without raising the toxicity. And so we are certainly getting lots of interest. As David said, we are on a path, and people want to see us roll that path out a little bit and get some more human results. We're about to do that. This is critical. This is a critical point for us right now. Those trials are critical, and in this regard, we are at the leading edge of this curve in terms of innovation in intratumoral type approaches to therapy. Michael, I wonder if I could ask you just your thoughts on this approach, the intratumoral microenvironment type approach.
I'm fascinated. I think the first time you spoke to me, Carmen, I was quite excited about this drug, and it's one of the novel things that I see a big potential for, and especially as you know that a lot of, like, only half our patients really get a good response to immunotherapy. So I think there's the other half that just have cold tumors, and that's an unmet need there. And so maybe a local tumor injection can sort of kickstart the immune system, as you talk. Like, once you get the immune system working, then you often see this abscopal effect where tumors that are not injected can start to respond to immunotherapy also. The other thing is that we see a fair number of patients that have locally advanced disease and they don't respond to immunotherapy. And it's a horrible situation if you can't do surgery. So if you have some of the bulky lymph nodes that you can't operate on, it's a horrible death for the patient. So having a localized therapy would be a huge benefit in these patients because you get better local control, which is important. So I think a local therapy is something that's really needed and is missing from our treatment paradigm right now. So
Yeah.
Yeah, that's incredibly important elements. Carmen, there's a question here in terms of, do you believe or expect we can achieve better efficacy relative to the initial monotherapy
Yes.
with our IGNITE and PRIME studies when combined with immunotherapy?
I mean, that this is what the preclinical research is showing us. What we expect to see significantly improved response rates because, again, and as Michael just said, what we're doing in the tumor and the microenvironments is activating immunity by exposing the cancer cells or making them visible to the immune system, and so that has to lead to better responses. There is a natural response rate. That immune therapy has capitalized on and done extremely well with. But as we've said over and over, it's plateaued. It's reached its ceiling. And so but the immune therapy is not over. These therapies are superb. They just are limited by what the immune system sees. And so we believe we can make it more visible.
Yeah, so I think it's fair to say that we're all extremely excited. I mean, everything we have done to date points towards tremendous success for these studies, and we couldn't be more excited. Of course, the whole durability aspect in terms of the contrast of what we saw before is, we were blown away, frankly, by seeing 8 tumors responding just with the priming function of THT. But it's really the durability aspect that is important, and that's what we're really going to see here, because we know THT primes things. Sure, that's what's now needs to be understood is just like that flat line curve Carmen showed before. Can we show that as well? Michael.
can I just say too, that just going back as an oncologist who's worked with immunotherapy since about 2005, 2006, is that these to have 8 patients who are resistant to immunotherapy respond to this treatment is really amazing because you just don't see those results. And the other thing is most of the focus now on immunotherapy is trying to, as Carmen says, trying to convert these cold tumors into hot tumors because tumors are very adept at making a very hostile environment to immune cells. And if you label these immune cells, like sometimes you'll inject these fluorescent labeled immune cells and you see them go right in the tumor, and other times they go to the edge, and it's almost like they're afraid to go in. They'll just stay on the outside. And so that's where most of research now is being focused on, is trying to convert these tumors into a hot tumor. So I think this is the type of drug that can do that.
think so.
Yeah.
I'm going to ask you a naive question, Dr. Smylie, if you don't mind, but you made a comment earlier that, you don't want to have too strong an immune response. Is there a fear that our THT therapy can have too strong a response?
Well, again, it's local therapy, so I wouldn't be afraid of a strong immune response locally. I think systemically, if you get too strong an immune response, we know that we can get a lot of toxicity to normal tissues. But again, it's a local treatment, as Carmen said. And so I think you're trying to limit the toxicity by giving everything local.
That's it. Wonderful. Well, look, thank you, Dr. Smylie. Really appreciate you taking time out of your schedule for us and to share these thoughts. And Carmen, we've just presented what we think is an extremely exciting future with these concept therapies in humans that have the potential to transform oncology. Thank you, everyone, for your time. We will keep you up to date. We're looking forward to kicking off these studies in the not too distant future, and we will keep you very much up to date on the progress as we move towards that and as we move through those studies. And we'll be excited to share, of course, the many milestones that come up in the months ahead and into the fall with the first early readouts. Thank you, and we're always available for you. Thank you.